Hello, this is class Bio 225 Genetics, and I'm Dr. Lucy Abkarian. We are going to review Chapter 4, Part 2 today. Somatic cells make the vast majority of our cells. For example, in G0's example is the neurons, which are not splitting, or there are splitting cells actively through meiosis. Example are hair follicles or the lining of gastrointestinal tract or bone marrow. We also have germ cells, which are the precursors for gametes, set aside from somatic cells during embryogenesis. So, for example, sex cells, the ones that are the only ones in our organs that are going through meiosis and producing haploid gametes. Here is an overview of meiosis, where a cell is duplicating the chromosomes, but then the cell itself is splitting twice, which causes on the end of having cell that contains only the half of the full set of chromosomes. Overview of meiosis I is the following. Homologs pair, exchange parts, and then segregate. Sister chromatids remain intact throughout meiosis I. Maternal and paternal homologs recombine and create new combinations of alleles. After recombination, homologs segregate to different daughter cells. And there are five substages to prophase I: leptotin, zygotin, pachytin, diplotin, and diakinases. Depending on the species, length of time in prophase I can be short or very long. Now let's look at those in more detail. So here is the graphical representation of these phases. For in figure 1, representation is for leptotin, where chromosomes thicken and they are becoming visible under microscope, and one can observe the movement of centromeres from the center of the cell towards the poles, opposite poles. And then in the second representation is the zygotin, where the final formation is happening, and the creation of the synaptonemal complex is taking place. And finally, the third one is representing pachytin, where the synapsis is complete and the crossing over of genetic information between a non-sister chromatid is taking place. So, and then it comes prophase I diplotin, where synaptonemal complex dissolves, and we can see already that the chromosomes, they look like something we used to see before. And then there is the diakinases, where the chromatids thicken and shorten, and also the nuclear membrane breaks down and the spindle begins to form. So then comes metaphase I, where the chromosomes are aligned in the middle of the cell, in the center of the cell, which is called metaphase plate. The microtubules are attaching to the centromeres to the homologous chromosomes. And then afterwards, cells go through anaphase I, where the centromeres don't divide, and then the chromosomes start moving towards the opposite poles of the cells. So we have to remember that meiosis I is a reductional division, and during telophase 1, the nuclear envelope is reforming, and the result is that cells have half the number of chromosomes, and each one of them consisting of two sister chromatids. And then there is the interkinases, where it's very similar to interphase with one important exception. There are no chromosomal duplications taking place here. In some species, the chromosomes decondense. In others, they do not. So meiosis 2 is very similar to meiosis 1, with only one exception, that even though we are seeing the cell division, we don't deal with DNA duplication here. Here is a graphical representation of that. Here is a very interesting example of prophase 1 of meiosis with very high magnification, where one can see the leptotin, zygotin, pachotin, diplotin, and diakinases with very high magnification. Mistakes in meiosis can produce defective gametes. For example, non-disjunction mistakes that take place, chromosome segregation during meiosis 1 or 2, may result in inviable gametes or embryos, which can be lethal, or they can result in abnormal chromosome numbers in viable individuals, which will cause different disorders that we have talked about already, such as trisomy 21, which is the case for Down syndrome, or XXY, Klinefelter syndrome. Many hybrids between species are sterile because chromosomes cannot pair properly. That's also an example with the mistakes in meiosis. There are two ways that we can get variations in genes. One will be the independent assortment of non-homologs that will cause the combination of different alleles, and also the crossing over between homologs will also bring variation. Looking at this slide and also taking under consideration what we have talked during this class and the previous lectures, please provide me with answer on what are the main differences between meiosis and mitosis. I will expect to receive those answers via email. In order to answer the question about differences between mitosis and meiosis, you can also use this slide as a resource. So by now we know that germlines are specialized diploid cells set aside during embryogenesis. Gametogenesis is the formation of the gametes that is involving meiosis with all of its pre- and post-meiotic events. Different type of animals have variations on general aspects of this process, as we have mentioned it before and shown on examples. In humans, for example, oogenesis produces one ovum from each primary oocyte. In humans, spermatogenesis produces 4 sperm from each primary spermatocyte. So here is a question. Taking under the consideration the material in your book, and you can use actually any resource you want to, please explain why is that so, that there is one ovum and 4 sperms. So now let's learn some terminology. Oogonia. It is the diploid germ cells in ovaries of female embryos. It divides by mitosis and enters meiosis 1 to become primary oocyte. Primary oocytes arrest in diploton stage of meiosis I until after birth. At puberty, one oocyte per month resumes meiosis. At ovulation, completion of meiosis I produces a secondary oocyte and first polar body. Secondary oocyte arrests in metaphase of meiosis II. If oocyte is fertilized, meiosis II is completed and produces a mature ovum and second polar body. So the interesting thing is that most of us probably knew all of these events, all of these by nature, but we did not know, but the terminology was probably unknown to most of us. Here is a question to complement the previous question regarding one ovum ratio to 4 sperm. Please explain also or provide me with answer, what is first polar body? Next few slides are for your observation and information. I'm not going to go into the details. So here is egg formation during the fetal ovaries and arrest during prophase of meiosis I, simply saying the ovulation process. Here is the description for spermatogenesis. As opposed to females, where the egg is developing during embryonic process, in male there is consistent development of sperm. Walter Sutton in 1903 stated that chromosomes carry Mendel's units of heredity in human, and here is the theory for chromosome theory of inheritance in humans. So it basically states that two copies of each kind of chromosome exist. Chromosome complement is unchanged during transmission to progeny. Homologous chromosomes separate to different gametes. Maternal and paternal chromosomes move to opposite poles. Fertilization of eggs by random encounter with sperm take place. In all cells derived from fertilized egg, half of chromosomes are maternal and half of them are paternal. So now we can apply Mendel's theory of inheritance to human genetics. So we have two chromosomes, two parental chromosomes with two of them containing two sister chromatids. Here is the matrix of all four chromatids, as each one of them can segregate into two. So then finally we are having the probability of 1 to 2 to 1 in the offspring that can carry information from paternal and maternal chromosomes. Okay, so in order to be able to break this down, it will be helpful to remember that two alleles of the same gene are located on the separate chromatids of the same chromosome. So now, if you take that under consideration, you can see the similarity between this, the human genetics, and what we were discussing during the Mendelian inheritance theory and explanation with big A, big B, small a, small b case. It's very similar to that. So prior to 1910, the chromosome theory of inheritance was supported by two circumstantial lines of evidence. First one was that sex determination associates with inheritance of particular chromosomes. True. Second was events in mitosis, meiosis, and gametogenesis ensure constant numbers of chromosomes in somatic cells. This theory confirmed and validated by inheritance of genes and chromosomes correspond in every detail, and also transmission of particular chromosome coincides with transmission of traits other than for sex determination. Here is an example of an X-linked trait in humans. That is the disorder where green and red differentiation is missing. This color blindness, in other terminology, is a disorder, the genetics of which is lying on X chromosome, which explains why men are more, like this is something that occurs more in men than in women, because women have second X chromosome, which will mostly mask it, where men don't have that second extra X chromosome, so there is nothing to mask it. I know we have talked about this previously, but here is the detailed schematics of an example of pedigree for an X-linked recessive trait. So this is an example of hemophilia. So as you can see, Queen Victoria was a carrier of hemophilia, and then Beatrice was a carrier of hemophilia during the second generation. And then between second and third generation, Alice was a carrier, and Leopard, offspring from Beatrice, had it. And then for the third generation, there was a carrier, and there was someone who had it, and then Alex was a carrier, and then fourth generation Alexis had hemophilia. On the right side, we had a carrier between third and fourth generation, and then Raptor had it. So let's pay attention on something very important. If you look closely, you will see that females are carriers, and the ones who really who have the disorder expressed are male family members. Here is an other interesting example of pedigree for an X-linked dominant trait, hypophosphatemia. So on the schematics, the square is male and the rounds are female. So let's separate these two cases and have a discussion separately. So for male, if male has it, his offspring, female, will have it for 100%, and male offspring will not have it for 100%. Now the case of female. In case of female, we have to separate two cases within. Case of heterozygosy and case of homozygosy. In case of homozygosy, both female and male offsprings will have it for 100%, and in case of heterozygosy, both female and male offsprings will have it for 50%. So finally, let's talk about autosomal genes and sexual dimorphism. So there are sex-influenced traits. Those appear in both sexes, but hormonal differences may cause differences. One of the very obvious examples is the male pattern baldness, so which is very, it's super common in male and very rare in females. And then sex-limited traits, those affect the structure or process found in only one sex. An example is Drosophila stuck mutant males can separate from female after mating.